Hello, everybody, and welcome. Just going to give a few seconds for any people joining just on the hour to get officially into the webinar room. Okay, well, we have quite a bit to get through, but we have budgeted plenty of time for discussion after today's presentations. I'm Iain Hrynaszkiewicz, Director Open Research Solutions at PLOS, and I'm your host for today's event. First things first, the webinar is being recorded, and with that in mind, please be polite and respectful of all participants. We do have a lot of people registered for today's session, so our plan is to only take written rather than spoken questions. And to that end, please put your questions in the Q&A feature rather than the chat. And if you want to share or chat on social media with others engaged with today's event, then please do that. And I would suggest using the hashtag open protocols, which should now be showing on the slide. All participants should be able to see questions and upvote any that you're particularly interested in to help us guide the things that we want to talk about following up the session. Okay, so that's the key points of housekeeping. I want to get on with introducing the session and introducing today's speakers. So we're going to start the session with an all-important researcher's perspective on sharing and reusing methodologies, giving us context on some of the real-world problems that we're trying to address. We'll continue with a closer look at the motivations for and opportunities provided by the protocols.io platform to meet some of these researcher needs. And we'll conclude the presentations with an overview of how peer review of protocols at protocols.io has been introduced in partnership with PLOS ONE to bring together some of the features of scholarly publishing with these more dynamic platforms for sharing methods. The presentation should take us about 30 minutes or so, and that should leave us a good 20 minutes for discussion. So feel free to provide your questions at any point during the discussions. What we're going to do is have all of the presentations and then all of the discussion afterwards, but please ask your questions as and when they occur to you. I just want to introduce each of our speakers before we start their presentations. Our first speaker will be Benjamin Schwessinger, who is heroically joining us at around 2 a.m. his local time in Canberra. Benjamin is a group leader at the Australian National University in Canberra. His team is mostly working on the genome evolution and molecular interaction mechanisms of pathogenic fungi and plants. Benjamin has a strong interest in open science. He is a co-founder of Reproducibility for Everyone, was part of eLife's early career group, and is a postdoc union steward at the University of California. Next, we'll hear from Lenny Teytelman. He is CEO of protocols.io. He did his graduate studies at UC Berkeley, and it was his struggle with correcting a published research method as a postdoc at MIT that led him to co-found protocols.io. Lenny brings to protocols.io a strong passion for open access, sharing knowledge, and improving research efficiency through technology. And finally, our final speaker, Emily Chenette. Emily is the editor-in-chief of PLOS ONE. She studied biochemistry at Columbia University as an undergraduate, earned her PhD in genetics and molecular biology from the University of North Carolina at Chapel Hill, and completed postdoctoral research at Duke University. Before joining PLOS ONE in 2018, she held editorial positions at Nature Cell Biology and the Febs Journal. While they're not speakers today, I also want to give a quick acknowledgement to Emma Ganley, Director of Strategic Initiatives at Protocols.io. So she and others in the Protocols.io team are in the background helping ensure today's events go smoothly. So we're very grateful of their contributions, but hope not to hear from them on the webinar because that might mean something's gone wrong. So just to recap, we'll have all the presentations sequentially. Feel free to post your questions as we go along, and we'll get to as many as we can. Speakers, just to remind you and remind myself that we'll turn off our cameras, mute our audio while the others are giving their individual presentations, and then we can turn our cameras and audio back on for the last part of the session. And final quick reminder that I am controlling your slides, so give me the pre-agreed indication when things need advancing. So with that, I am going to move on to Benjamin's first slide. And
all right. Thank you, everyone. Thank you for having me. So yeah, it's really early here. And before I start, I would like to acknowledge the past elders, emerging and present and future of the Ngunnawal people of the land where I'm come from, and maybe you can do something similar from if you're in a country which does acknowledge its elders, emerging and future. So think past, present, and future. So thank you for that. So I might maybe now go into why and or would you or I care about robust and reproducible science. So maybe start with a slight personal touch here. If you can go next. So when I did my postdoc, which was at the University of California, Davis, I went to a big lab, as many people do, to try to get like the next big paper out. So this was about a big science paper at the time. And when I arrived, I was really like, is this actually all what we have here reproducible? Can we actually build on this and so on? I'm not going to go into like big details, but me and others, we were really confused about it. And this was then, we worked through this quite some time, for 3 years really, where we couldn't reproduce these findings. And my boss at the time, Pamela Ronald, she really did a great job like working with us through these problems. And she wrote, for example, one article like this in Scientific America about how a retraction works and an anatomy of the retraction. Instead of like brushing under the table, she really went out there and corrected it, which is a great achievement, I think, for her and for us too. And so I just mentioned here on the slide a couple of issues which we ran into, which is mislabeled stocks. We had a lack of detailed protocols. That's probably why I'm here today, and also insufficient methods. And what really came to my mind, looking back at this, is this fourth law of thermodynamics, which is of Twitter, of course, like many things. And this is really the amount of energy necessary to refute something incorrect is an order of magnitude bigger than to produce it. And if you have published a big papers, like this paper, which we retracted, was in Science, it was quite an effort to actually figure out what was wrong, why it went wrong, and also what is the correct thing. So we managed this at the end, but it was also a lot of energy which just went a little bit down the drain, or maybe could have been used for something else. And then it took us on a couple of years later. I moved to Australian National University of Ngunnawal people land, and I considered just a little bit what I could do when I started teaching here, how we could do maybe some things better, and also how could we maybe pass some of what we learned on. And so we started a course which is based around scientific rigor, and part of it is about experimental design and error analysis and statistics and so on. But we teach basically note-taking and have a scientific process a little bit here. And the overall idea is that really that nobody's perfect. Everybody makes mistakes. We're all learning here. it's important to start this journey of robust and reproducible science early. So we teach this at the master's and honors level. So just when people finish the degree, and then also at the PhD level too, when people start. And the end goal is really to give people ideas about transparent science and open science, and really so that you can actually report science, how it was done. And because if you do that, somebody else can actually read it and make their own judgment and say, oh yeah, I believe you not, or I will build on yours, this or not. I think that's really the end goal. It's not so much about being right or wrong. It's really about being transparent and open. And one of this is about protocols, of course, and we do this allegory of drawing, and then you can maybe step me through this now, Iain, slowly. So I ask the students to take out a piece of paper, next, and then everybody should imagine a beautiful owl. The owl is a nice concept because it's pretty inert to cultural differences, so everybody really knows an owl. And then I just give them the exercise to just draw one. Don't share it. And then the next step is to write these instructions down. So little similar that you would write a protocol. This is then shared with another partner in class. The other partner has to draw this up on another piece of paper. And then we share this before and after with class, like within the class. And I think it's a great exercise because it's a simple, but it also really shows that even a really simple concept like draw like an owl, it's really, people draw this differently. People take different things away. And also, it gets initially sometimes a little lost, is that people really describe things very, very differently. And people also read things and use instructions very differently. And then it's always really fun because everybody laughs about each other's owl. And some people actually just pin it up in the office and like have this motivation too. So it's a good laugh and people like enjoy it and get the point. And it's a little bit like this. So often a method section is like, oh yeah, we did this amazing experiment where we, in biology, like petted 50,000 liquids in a tube and spun them 3,000 times at really different speeds. And then we took like a tiny piece of it and measured it in a certain instrument. But this is really amazing. And so similar, if you draw an owl, often the instructions are very scarce, and really what the authors want you to do is to draw a really perfectly nice owl in a certain way, which probably looks like only the owl in their home village. All right, so after we get this home, we really got into the scientific process of record keeping. So actually before the protocol is actually there, I will come to this in a minute. And then I asked a question around if people actually know how to take good records. So what's part of a good record? And then depending from what class they took, some people say yes, mostly say no. And also like a completely unrepresentative survey on Twitter reveals the same things, that most people actually have very little training in record keeping. And that makes you maybe also not, that isn't then too unsurprising that protocols and sections and methods might not be that great if you don't even know how to record it. So why should we actually keep a good record? It's because it's the step before the protocol. And this is from a paper which I can't find anymore, but I think it was a really nice introduction to this, is that taking records is really the start of it, because it's an information synthesis. And then it becomes a formalization. If you do an experiment multiple times, you optimize it and you formalize it. And then really the protocol at the end, which you share, is the tool making. So hence record keeping is really such important to share and build science as part of a tool making process. So what also people, especially students, often forget is that the initial information synthesis really trains your thoughts. at the other end, you have the tool making that really enables communication. It's important, of course, simply to record what you do. And then I have all this compliance and IP issues, which is part of formalization. And really what we're talking today about is really about enabling collaboration and bringing people together with sharing these protocols out once they are a tool. Okay, yeah. All right, so, which is really bad screenshot, I apologize. This is one of protocols.io, and Lenny will introduce this a little bit more. So this is the tool making side of the proposal. It's really protocols.io kind of felt as an empty void where people could simply share the protocols out. And the first year of it, I think 2014, 15, just when protocols.io started, because Lenny invited himself to University of California postdoc event. We had some other PLOS and eLife, and so presenting to the postdocs at the time. And then I took this up and I went to Australia, and I really liked it because I liked the idea of publish first, review later. I know when my protocol works, I know when my work is kind of ready to go out, so people can just pick it up and use it. So we started it in 2016 to put some protocols out into the wild, and we mostly focused on genomics and the downstream part of actually getting long DNA out for the sequencing. And I presented this in several workshops, and people were like, what is this guy talking about? It did work out at the end, so you can't really see this here, but what I circled in blue is that we have like 50 protocols on there. We have 500 members in this group now. We have like over 100 discussion group and points and 13 resources. Across the group and a site, which is like protocol, we have 60,000 views. And I know in person that we accelerate the science of many, many, many people. And the funny thing about protocols.io is that more people emailed me or asked me questions about my protocols than on any of my publication ever, which is kind of interesting. And then I would just like to highlight a couple of protocols which we have. For example, that was from Josh Quick. I really appreciate that Josh and other people in the long read community contributed early to this. They liked it, and so for example, this specific protocol has like something like 40,000 views, which is quite a lot for like a random protocol or a random web page. Lenny, no offense, just out there. And then I think it was also great that Josh joined this, because also Josh and others did develop prior to COVID viral genomic sequencing for Ebola and so on. And they were ready to go just to publish this protocol, this specific COVID SARS-CoV-2 sequence protocol, apologies, SARS-CoV-2 viral sequence protocol, which has over 100,000 views. And this was out really early on, like March or maybe even earlier last year, like really early. And it was great to have a platform where people could just go and everybody could just pick it up and ask questions, which you see on the right here too, a little bit. But Lenny will probably go into this. So I think it was really great, a platform simply to share protocols if people knew about it. So thank you really much for all the contributors to our specific group and really anybody else on protocols.io, because I think it does accelerate people's science if you know about it. And that brings me really to my endpoint here, which I would like to make. Of course, it's great that students learn some information synthesis and formalize it and make a tool, and then put a tool out online, like protocols.io. But the next step is really the distribution and the uptake. Because, for example, I put personally a protocol up, one of it in 2016, which is also about long-read sequencing, DNA from fungi mostly, because it's what we work on. And people who found it really loved it. It's really good. And I also presented this with reproducibility for everyone on different workshops. And then often also the question which came up like, why do you would encourage students to put something on a website which nobody knows about? More and more people know about it. Like, it takes a long time to get anywhere. But so maybe flip the question on its head. It's really, how can we make a protocol.io even more findable, so to speak? And also the reward system came up. So why would a student put something up there if they don't get a citation for it? Why wouldn't you publish it somewhere else? And that's what we did actually with our early protocols too. So we actually put it online, and we also published it as a book. As you can see, another advantage of protocols.io here is that it's actually for free. I personally can't even access this book without certain nice web pages, which you can use sometimes. But it's really addressed a challenge, I think, or there was a challenge that you had this tool making in the process. You had a great thing on protocols.io. People really used it. And that where plus one comes in, I think, really nicely, is that you can put now your protocols on protocol.io. You can publish it whenever you like in such a way. And then people can peer review it. So it's published first, peer review later a little bit. And people maybe already used your protocols hundreds of times, but you still get the reward at the end if you get published with +1, and also takes care of the distribution uptake, hopefully over the years. I always use this too. It's because it's findable in PubMed, probably also slightly better in Google and so on. And then people hopefully also go more to protocols.io. So we actually just got our first paper on this pathway accepted yesterday. So it was really exciting. It will be out soon with one of those protocols, which probably has been used quite some time already. And the research officer who did it has a first author paper now. So for his career, it is quite nice to have this too. It's also corresponding author. And what we started now in the lab is that we will have one of our protocols converted every half a year. So we will have always a small competition. And then we will publish and convert one of our protocols.io papers, publish in a peer-reviewed journal after. And I think it's a great new way to get to this publish first, peer review later, and then publish in a peer-reviewed journal. So that's why I'm really excited, because it will make it more accessible, findable, and hopefully encourage many more people to share the protocols and protocols.io and then later on +1. So thank you for that. That's all I had to say, and I'm happy to take questions later.
Thank you, Benjamin. We'll move straight on to Lenny's presentation. Just before we do that, quick reminder, if you have questions, if you click the Q&A button at the bottom of the control panel, put your questions in there to make sure that we get to them. If we do see any that didn't make it, that are in the chat, we'll do our best to move them over. Okay, Lenny, over to you.
Thank you, Iain, and thank you, Benjamin, for your section. It makes it very easy for me to present. Before I start, I should also just as one item of note, we shared the link to the slides that we're showing in the chat. We'll also include it in the email that goes out to everyone who registered with the recording. Any of you who have tried to access it a couple of minutes back might not have had the permissions to access, but that's now been fixed, so you can everyone can access the slides and then reuse them, just like all of the materials and reproducibility for everyone workshops that Benjamin talked about. So Benjamin did what I always do in my presentations, which is talk about the importance of methods and the challenges with sharing the details and reproducing work from other people's labs or your own lab. So I don't actually have to talk about it. I'll just go very quickly over some of the key features that actually Benjamin also mostly highlighted already. And then I'll talk about why, from our perspective at protocols.io, why the partnership and the ability to peer review protocols is so important. So the mission of protocols.io is to make it easy to share method details before, during, and after publication. And as Benjamin said, we launched in 2014, and Benjamin was one of the earliest people sharing on the platform and pulling people by their ears and highlighting the importance of sharing the methods. And you can see that we are an open access repository with over 10,000 public protocols, but there's also a private site for companies and academic labs to organize their knowledge internally. So there are over 30,000 private protocols and 10,000 public ones. The business model at protocols.io, just as at GitHub, is if you're sharing publicly, it's open access, CC BY, free to read, free to publish, as Benjamin said. And we ask for subscription if you're using it in private collaborative mode within the organization. And next slide. So this is if the video cooperates. And of course, it will need to be restarted because nothing ever cooperates in technology. Paper copies are sometimes nice, despite us encouraging digital. The protocols are dynamic and interactive. They're not just static PDFs. You can ask a question on any step that'll go to the author and everyone who is using the protocol. You have robust discussions and Q&A that builds up. We have materials section. We try to work with reagent vendors to get vendor catalogs to make it easier to document exactly which chemicals, kits, and equipment were used. There are DOIs minted for every public protocols. Everything is preserved and preserved in clocks, mirrored in Internet Archive and on a GitHub repository. And what you can see in terms of what we just showed in the video, right, that there's also structure to the steps. It's not just text, and I'll talk about that in a second, so we can actually go to the next slide. Thank you, Iain. So there is a very powerful editor that we've put a lot of energy into before we launched and in the years since 2014 to make it easy to capture whether you're sharing computational workflows or experimental or field methods, to make it easy to capture the details that are necessary for someone else in your lab or a reader of a paper that you publish to reproduce your work. So as I said, we try to get catalogs from reagent vendors, try to encourage people to explicitly share the software that they use, which version, which parameters. If you're talking about a data set, what version of the mouse assembly was it? So capturing those small but important details. All of the protocols, they're not passive tense paragraphs with vague references to we did it somewhat similarly to these other papers. We try to encourage step-by-step reproducible reporting. There are more and more protocols, including some from Benjamin, that include videos. So with this amazing phone technology that we now have in our hands, everyone can go into the laboratory, and if you have a complicated step, take a 10-second, 30-second video, put it on YouTube or upload it directly into protocols.io, and that video will render right in the step. And the videos are a really important component of sharing things that across language barriers. And even if you are an English speaker, it's sometimes hard to read just text, and seeing a video of how you're doing the sample preparation, how you're handling the tissues, can make all the difference in trying to replicate the work. Just like Google Docs, there's concurrent editing, so you can work with collaborators. And commenting, we already talked about, that you build up an FAQ, and instead of answering a question over and over, the same question by email after you publish a protocol, you can just answer it once on protocols.io, point people to it, and hopefully over time people stop asking that question because they see the answer right away. And next slide. So this is just a view of what some of these components are that I mentioned, and what that structure is. And the structure, there is a reason why we try to encourage, there are several reasons why we try to encourage not just text inside the steps, but the images, videos, and the components. It improves readability. And when you version, when you make copies of the same protocol, it becomes easy. I'll show in a second. It becomes easy to compare the different versions if there is structure, so we know that you changed the amount or you changed the script that you were running. It makes it very easy to detect what are the changes between the variants of a protocol if there's structure inside the steps. And if we go to the next slide, there's another reason for components. As I said, about 5 to 10% of the 10,000 protocols in Protocols.io are computational and bioinformatics workflows. But for the experimental wet lab methods, there is a run button on every protocol that allows you to step through it, sort of like a checklist cooking app that allows you to step through the protocol as you're doing the work. And that's where those components amounts play a role, not just for readability, but if you took the time to put in those components, then when you're running the protocol, if you have the timer in there, you can start it right on your tablet in the Fume hood without just printing out a PDF copy. You have amounts that you can scale. So if you're doing 10 times the samples, you can just multiply all of the amounts across the volumes in the protocol. So there are, these are just some of the features of Protocols.io that hopefully not just make things more reproducible, but save you time as you're using the protocol. And next slide. And one of the most important things that comes across that owl drawing exercise that Benjamin mentioned is the importance of having versioning for methods, because unlike a paper, which is a snapshot in time, these are the results we got, protocols evolve, right? They're sort of dynamic, living, breathing creatures, and we constantly tweak and optimize. And as I said, part of our mission is also having a place where you can not just report the details at a point in time, but can come back and share corrections and optimizations. So that's particularly important for methods because they tend to rapidly evolve. And while we preserve, just like any publisher, we preserve anything that's published and has a DOI, you can't just go back and change the protocol that you shared. You can easily, with a click of a button, especially based on feedback and questions and answers that you have, you can easily make a new version of a protocol, and then everyone can still access the earlier version, but is automatically guided to the most up-to-date. And next slide. Not only do we have versioning, but for those who know GitHub, there is forking. So every protocol sort of serves as a template, and the author can keep a protocol up to date through versioning. But if you're working with liver tissues and I'm working with lung tissue, we might have slight tweaks and variations, and every protocol serves as a sort of template, public and private protocols. You can create a fork, and Emily will talk a little bit more about it in the beautiful example of the first paper that just came out as part of the Protocols +1 initiative. But the forking allows you to share variations and then get credit while preserving the link to the original and trying to give credit to both the original authors and the new authors of the fork. It is similar to versioning, but allows you to share other variants of the protocol. And in this example that Benjamin already showed from Josh Quick, the protocol on SARS coronavirus sequencing, this was the very first protocol on Protocols.io shared on coronavirus sequencing back in January of last year. And they actually, you can see at the top under the title, they made a fork from a protocol they shared several months before that in September of 2019, before COVID-19, for how to sequence Ebola. And for both versions and forks, if you go to the end of the title, there's a compare button, next slide, that allows you at a glance to see what's the same and what's different between the forks and versions. So in this case, on the right is the Ebola protocol. On the left is the coronavirus sequencing protocol. And you can see literally that, you know, in step one, the change is instead of 10 microliters of Ebola RNA, you need 11 microliters of SARS-CoV-2 RNA. And these are the things that the forking is meant, these are the things that the forking is meant to capture. And the compare button makes it easy to see. And I'm almost done, so let's go to the next slide. So I talked, and Benjamin also talked about a lot of the features of Protocols.io that make it easy to share these details. But the thing that we were missing since the very beginning that I was dreaming about is the ability to peer review and get formal credit and improve the recognition and credit for the painstaking work of method development that researchers put months or years into. And Benjamin had asked me, I think in 2015, for suggestions of where do you formally publish protocols. And here is a 2018 example of a professor, Professor Avello, saying that at a meeting, topic came up that PLOS should start a protocol journal to publish standard operating procedures and protocols whenever useful, not just at high impact. Somebody else said, well, couldn't you just do it on protocols.io? And the professor said, yes, but you need peer review, you need traditional citable journal publications. And I, next slide, and I think that's my last slide, and I periodically on Twitter will answer, yes, you're right, we don't peer review, we're a platform, we're not a journal, and yes, you do need the ability to peer review. We can't do it at protocols.io because the scope is so broad. You kind of need to build PLOS ONE to have thousands of editors in order to be able to handle diverse papers and peer review them and turn them into journal publications. And that's precisely where PLOS ONE comes in. And this is why we're so thrilled about the new initiative. So my part is done, and Emily will talk about that.
Thanks, Lenny. Emily, over to your presentation.
I am absolutely delighted to be here this afternoon. And Lenny, thank you so much for that really amazing tour of all of the wonderful features of protocols.io. And as you said, it's exciting to work with us. We are delighted to have the opportunity to work with you too and bring this to the scientific community. So a little bit about PLOS first before I launch in. So PLOS launched in 2001 with a mission to accelerate progress in science and medicine by leading a transformation in research communication. 20 years later, our goals haven't changed. We're still committed to breaking boundaries by advancing research faster, sharing more broadly, increasing collaboration. We want to empower researchers to transform science through choice, inclusivity, credit, and transparency. We're trying to redefine what we mean by quality. We want to assess and communicate the full arc of research more fairly and more accurately. And finally, and very importantly, we want to open science so that we contribute to the building of a foundation of knowledge that helps move us forward. Next slide, please. So in 2006, PLOS launched PLOS ONE, and this journal has a key role in supporting PLOS's mission. It has a broad scope, so it publishes in all academic areas, including multidisciplinary studies, interdisciplinary studies, and null and negative results. We have 7 unique, well, at the time they were unique publication criteria, which mean that we focus on rigor and methodological and ethical rigor rather than the perceived impact or the perceived importance of the work. The journal also drives open science practices from support to preprint. We publish the peer review history of manuscripts if authors opt in. We have a very robust data availability policy, comprehensive reporting criteria that's assessed at several different stages during the review process. And we also support material code and software sharing. The journal also aims to support researchers throughout the research life cycle, so not just one single manuscript submission. We hope to offer authors credit and transparency from the design to submission to publication and beyond. Next slide, please. So this last point that I just mentioned, this credit and transparency in study design, is part of what fueled the wonderful partnership with protocols.io. So Lenny alluded to this in his talk when he mentioned that protocols.io is a platform that provides open access to protocols but doesn't peer review them. So why might peer review be important? So let's look at some of the barriers to this first. So we have citation trails where papers are stuck behind paywalls, and so or the methods aren't reported in detail, so it makes it very difficult to understand and to reproduce studies. It's also possible that the method sections may not be detailed enough to reproduce the paper. This can also have knock-on effects on the reliability of the conclusions, especially if the paper cannot be reproduced. There are also some clear benefits to publishing research methods, and this includes that the opportunity to get peer review feedback before a study is started. So this can help lead to more consistency and more rigor in the research project. We also recognize that the process of developing a protocol is complex and demanding, and not everyone that contributes to protocol development may get a first author paper out of that experience. So we strongly believe that researchers deserve credit for the work that they do, and that readers will also benefit from this knowledge in protocol development. Next slide, please. So to meet this need, PLOS ONE has developed a suite of options for authors who want to publish their protocols. We have registered report protocols and registered reports that are two independent publications. It's the protocol that is reviewed, and if the protocol is reviewed favorably and published, the authors receive an in-principle acceptance for the resultant study, and that's the registered report. We also have our standard research articles, and authors are free to contribute their methodology as a standard research article that's peer-reviewed and published. Study protocols are an article type that launched earlier this year, and these describe plans for conducting a research project. So these have to be published before the research project is completed, allowing for peer-reviewed feedback before the work is started. And then finally, and relevant here, is we have the lab protocols. And this is a pair of peer-reviewed and interlinked components. So there's the step-by-step protocol on protocols.io, and then an article that's published in PLOS ONE that offers additional contextualization to the protocol. Next slide, please. So lab protocols are formally peer-reviewed, published, and indexed. And here's what that looks like. So authors have the opportunity to submit directly to PLOS ONE, or if they've already posted a protocol at protocols.io, they can transfer their submission to PLOS ONE through a nice interface. We offer support for authors who have not yet posted their protocol on protocols.io. It's very easy to upload protocols on protocols.io, but if people need additional support, we're very, very happy to support authors in this process. So once the protocol is submitted to PLOS ONE, it undergoes a series of internal checks that are handled by in-house editors. After it passes the internal checks, it goes on for editorial and peer review. And so one of our academic editors, our board is about 10,000 strong now, and that academic editor will invite one, maybe sometimes two additional peer reviewers, just to make sure that the methodology appears robust and that it's reported in sufficient detail. The authors will then receive the editorial decision, which in most cases is a minor revision. We'll go back for review, and if the authors haven't already uploaded their protocol at this point, they can upload to protocols.io after that first editorial decision. Once the academic editor and reviewers are satisfied, the manuscript at PLOS ONE is published, and the protocol at protocols.io is released if it's not already published. Next slide, please. So once published, lab protocols contain several features that make them a really unique and exciting article type. So on protocols.io, and that's on the left side of the screen, I think it's left side of the screen, we have the title, the interactive features that Lenny showed so beautifully in his talk, and then finally those step-by-step interactive instructions with the measurements, reagents, and more. Then over on PLOS ONE, we have the title, the introduction, the method section, which is usually just a link to protocols.io, and also the expected results. And so these sections that are published in PLOS ONE act as a very nice wrapper or container that offers additional context to the article that links back to protocols.io, which offers the protocol in all of its depth and in all of its detail. The protocol is also publishes supplementary information in PLOS ONE, in case authors would or readers would like to download or print a PDF to have with them in the lab. Next slide, please. So here's what this looks like in practice. This is the very first lab protocol that we published in May, just over a month ago. So on the left side is what it looks like in protocols.io. And part of this interactive feature here at the top is the ability to indicate whether or not the protocol works. So it's kind of this post-publication review, so that there's additional opportunities to show that the protocol achieves its intended purpose. And then in PLOS ONE, we have links back to protocols.io, and then what looks like a traditional research article with the badge at the top indicating that it has been peer reviewed. So next slide, please. So what are some of the benefits of publishing protocols for authors? Well, we think that it helps ensure reproducibility and increasing trust by sharing detailed step-by-step protocols. So it means that the experiment that you've worked hard to troubleshoot can be used by others to support their research. It also enables people to receive expert feedback through peer review to help refine and shape the protocols, either before and then also after publications. Lenny mentioned this versioning and also forking pathways on protocols.io. It allows authors to get formal credit for protocol development in this peer-reviewed and published article. And it also increases the visibility and discovery of protocols through the indexing and archiving process. Next slide, please. So finally, I just wanted to share some of the wonderful feedback that we've had from the author of this very first published lab protocol. His tweets here so beautifully capture some of the reasons why we're so excited about this. So he mentions that it's his first author paper, that it's a newly updated protocol with an extensive background that provides the rationale for methods. And I just love this part. He says it's an awesome and smooth system for sharing his research. Next slide, please. And finally, another really exciting aspect of this first publication, which again, Lenny alluded to in his talk, is that the protocol on protocols.io has already been forked, and another researcher has used that first protocol to develop an approach that uses the vector graphics editor Inkscape to do the analysis. And it's just something that I think is really lovely, really exciting to see this use in the community. And that's it. Next slide, please. I guess I can take this opportunity again to say thank you so much for spending time with us today. And I guess, Iain, if I'm not stepping on your toes, we can open it for questions.
We can indeed. Thank you, Emily. I am going to turn the slides off in a moment because it gives everyone a bit more room on Zoom, which I'm sure we're all familiar with in recent months. So just quick recap. If you want to join the conversation on Twitter, it's #OpenProtocols. So going final time, I'm going to stop sharing my screen. Get myself organized. So we have everybody has their cameras on. We have a few questions, and I can see Benjamin, Lenny and team, you've already been hard at work answering some of the questions. So good to see that happening in parallel. The first question I think is for Emily, and it's in relation to study protocols that you mentioned. The question is, what kind of study protocols can be published in PLOS ONE?
Sure, that's a question that I'm really happy to answer. So the answer to that is anything that is within scope of PLOS ONE, and that is any academic research. We have our publication criteria. We have 7 very discrete criteria by which all new manuscript submissions are assessed. But in terms of the scope, it's wide open, from physical sciences to life sciences, social sciences, and public health and medicine as well.
Thank you. I'll move on to the next question in a moment, but what I will do is I'll just give a quick summary of the answered questions if we have time for people who are listening live. I think the next question is also for you, Emily. But if Lenny, Benjamin, you have opinions on this, feel free to throw them in as well. So the question is, are the editorial and/or peer reviews for lab protocols via PLOS ONE blind, or are they open?
So the peer review and the editorial review operates in the same way as they operate on all other manuscript submissions. That is that the editors and the reviewers know the names of the authors of the submission, but the authors are not shown. They know the editor once the decision is sent, but they're not shown the name of the reviewer unless the reviewer opts in to sharing their identity.
I'll assume silence, no urgent opinions from the other panelists on that question. This one is, the question is, what about an update of an existing protocol? So why don't we take the perspective of Lenny, how you deal with updates, and then, because this could be applied in different contexts, and then see what Emily has to say, or Benjamin.
That's actually similar to a question that's already answered as well. So as you go over the summary, it touches on it, that this really is at the heart of one of the unique features and important features of not just having it in a journal, because the journal provides the important snapshot that Emily talked about, the peer review, improving the quality, but the methods do evolve, as I said. in my part. And so the paper, like you would expect it to, has to remain fixed for obvious reasons for scholarly record. But because of the link from the paper to protocols.io and the easy versioning capability on protocols.io, that first version never disappears, but we hope and look forward to the authors coming into protocols.io, answering questions like Benjamin was talking about, and then improving their protocols, making them clearer, right? So the paper is the paper, but the protocol can continue living and evolving. And as new equipment comes in, maybe you improve it even 2 years down the road, right? And that is very much at the heart of this initiative, to have both the paper and the ability to update and share the tweaks and optimizations.
I just thought it'd be interesting because it's a fairly broad question. Benjamin, in sort of in the real world, how common is updating of protocols? Where does that fit into your workflow in your lab?
I think the first protocol we put up, I think we have 11 versions. So it did evolve quite a bit. But that's my personal take. That was one of the personal protocols I put up. And I'm a little lazy initially. I just want to get it out, and then I refine it over time. And also with the question, I think it's really useful. And then we added also to this one, like videos, because people always got stuck on a specific step. And so videos, I think, are great for that. Also for maybe non-native speakers, I think that's also, and for native speakers, but like also for non-native speakers, like myself, really. And yeah, I think so. I think it's a really important process, and it's really nicely done, I think, personally, in protocols though. And I think when I was asked initially about how PLOS ONE could actually handle this with protocols.io, I was really adamant not to actually replicate the protocols.io feature of versioning, and that the protocols which is linked out then can be still live and evolving, because it's such an instrumental part of science. And I think that's actually quite nicely done in the partnership.
Okay, Emily, finally, how would you answer that question editorially in the context of updates and how you would view those at the journal?
Yeah, so before we began publishing lab protocols, we have a history of publishing methodology papers. And so most of what we publish, I would say, are updates or refinements of existing techniques. And so the journal is wide open to considering these sorts of publications. We do have criteria about validity and about utility. And so we would be asking our editors and reviewers to make a judgment about whether or not the new method, that the improvement is a sufficient improvement over the existing method that it warrants a new publication. But generally speaking, we want to see improvements and refinements out as part of published literature.
Thank you. I'm going to skip to another question, which I think relates to scope, actually. So it seems quite pertinent for today's discussion. Someone has noted that at PLOS ONE, we mentioned linking in the introduction section to at least one supporting peer-reviewed publication in which the protocol was applied. And they're asking, does this mean the protocol has to be submitted at protocols.io first, get some citations at other publications, and then be able to be considered in PLOS ONE? Perhaps you could clarify that, Emily.
Sure. So this is a relic for kind of how we first considered approaching this project, where we thought that to kind of distinguish lab protocols from other publication types that were out there, we wanted to make sure that this validity requirement was met right off the bat. But after thinking about it some more and considering the feedback that we'd already had and seeing what wonderful things can come of this partnership, we decided to open the criteria so that now all research methodologies are in scope as lab protocols. So we have the research articles and lab protocols, and they're very complementary. So we're giving authors the choice for where they feel that their research best belongs, whether as a lab protocol or as a research article.
Any other comments on that one?
So just to be crystal clear, Emily, I don't think there is a requirement that there is a link to the protocol in some other paper before submitting to lab protocols. Is that correct?
That's clear. Yeah, that's correct. And we're in the process, because this decision was recently made, we're in the process of updating all of the information on the website. But it's clear we haven't yet done 100% of it yet, but it will be updated very soon.
Okay. Couple of other questions here. Seem like more of comments, more of a comment than a question, but thank you for those, everybody. I am going to just give you a quick summary. So there was a question that was answered live that was thinking about encouraging non-native English speakers to make contributions, suggesting, for example, multilingual protocols. Lenny, actually, rather than me fumble through it, do you want to maybe just give a quick summary of how you answered that question about multilingual protocols?
So what I said was that we very much think of the global audience, and only 30% of the traffic to protocols.io is from United States. And that's why, as Benjamin said, we encourage videos. We think that it helps sort of this. We don't have the capacity to translate 10,000 protocols into different languages, of course, but videos are a natural localizer. Even if you don't speak perfect English, it's much easier to follow a video than just text. And in terms of language support, I put a link to the FAQ on protocols.io. We do ask for an English version, but because of the global audience, we warmly welcome translations, right? That can be a fork of the protocol. We warmly welcome translations of the protocols, and we do have translations into Japanese, into Portuguese, Spanish, and other languages of protocols that the researchers themselves provide, and we are always thrilled to see that.
Thanks. We've had another question come in again about scope, so I'll try and answer it to see if to see if my perspective answered that. So someone's again commenting on the publications criteria for lab protocols. So we publish methods which lab protocol articles which have been used in a previous publication, or they can be a modification to a method that has been used in a previous publication. And we're glad to consider those, as well as new methods described in lab protocols. And in those instances, because one can't link to a peer-reviewed publication, we ask that the author provides data demonstrating that the lab protocol has been used. So they're open to previously published, new and modified methods. We are very nearly out of time, but there was a question, Benjamin, which you answered, which I think is on a different topic, and let's quickly cover it if we can. And that was, what are some tools used for record keeping? To what extent do electronic lab notebooks cater effectively for this, or is it more of a human training problem?
Yeah, so I did provide some resources. So the Harvard Library has a good rubric for what different electronic notebooks are out, and I think I pasted this in the link also. If you do Google Harvard Library and electronic notebooks, you find this, because I found it right now. And also be it reproducibility for everyone, have a little bit of section on this. So you can find the different ones, and you can buy your own flavor, or like take free flavors if you like. If you take free flavors, do be worried, or at least read the IP. So actually who owns it. And I think to the usage, I think if you have new students coming in your lab, I never had a problem picking up any electronic notebooks, because it's just what the younger generation grows up with. So I don't think that's really an issue at all. It's just more, slightly more senior people than myself, maybe, which still likes to write stuff in hand. I also wanted actually to chip into this previous questions around how the publication process works, because we just went through a publication process. And with PLOS ONE, we also published a protocol, which I think as such was not part of a previous publication. It hasn't been cited, but it had we generated lots of data with it already. So that was sequencing data. So for example, we partially referred to just short read archive data. So that, oh yeah, people actually used it when we deposited data, or we simply made a small figure out of the results, so to speak, which didn't really reveal anything from the publication of the bigger scientific finding, but did show that the protocol works. And so I think that's actually also a nice additional feature that I think people are more encouraged to actually show that your method really works, and maybe a little bit preliminary results around it now with PLOS ONE, which protocols are you can do but don't have to. So I think actually I like this encouragement, kind of a little bit of a mini benchmarking within the PLOS ONE framework.
Thank you. And with that, I am going to have to draw today's webinar to a close. We're a minute over, so apologies if people have other meetings to get to. I just want to say thank you very much to everyone for listening. Recording will be shared. Slides have been shared. Hopefully this has been useful for everyone attending. And thank you very much indeed for the speakers. Thank you, especially Benjamin, for joining us at such an unsociable hour. And with that, I wish everyone a good day. Thank you. Goodbye.